Okay, so we're going to be doing the CXC Human and Social Biology crash course. So basically, we're going to be going through a section of the syllabus right now. Let me just make sure that the sound is up. Yep, so that's it. Okay, so yep. So basically, we're going through this crash course of some topics within the CXC Human and Social Biology syllabus, and basically what is not mentioned in here, you'll be able to see the reproductive system, cell division, meaning mitosis and meiosis, hereditary and variation, that's coming up soon, and also the nervous system and which other one? The nervous system, reproductive system, and all of that stuff. So we can just leave that to the side, and you guys can just go and look at those down below. But yeah, so let's just get started. You can obviously go ahead and check out everything that's going on right here. So basically there is a CXC study guide, right? So the CXC study guide just basically giving some tips on how to prepare for your CXC exams. There's also I think there's a video with the best CXC textbooks, meaning like the recommended textbooks, stuff like that. So you can also go ahead and look at that, and obviously tutoring right there. So yeah. Okay, so as I said before, if the reproductive system, you can be on the lookout for that, or you can look down below. That's the reproductive system, nervous system, cell division, meaning mitosis and meiosis, also cell structure and function. Cell structure and function, that's a separate thing. Cell transport. So all of them will be down below, and you can go ahead and look at those on your own time. But for this session, we're going through these 6 main areas, right? So obviously we're going to be looking at characteristics of living organisms, different methods of transport across the cell membrane, talking about some microorganisms. We're going to be talking about bacteria, fungus, and virus. No, virus is there's a reason why the virus is in red, because it's debatable whether or not the virus is a microorganism or not, but whatever. Then we're going to be looking at cell structure and function, photosynthesis, and food chains, and nutrition and food webs. Yeah. So anyways, let us just get started. Okay, so if you're studying biology, you have to know this. You have to know this. So basically, the characteristics of living organisms, and this is literally, if you have the CSEC HSB concise book or the CSEC biology concise, you will see that I think it's on the first page, in the book where they start with cells and stuff like that. They have the characteristics of living organisms. So basically, what that means is that if you're studying biology, you need to know what constitutes to be a living organism, because obviously that is what biology is, the study of living organisms, right? We don't talk about dead things. We talk about everything that we talk about in biology is 100% living. And if you're doing a subject, you need to know what constitutes as living and what is not living. So even though this is HSB, you still need to know, right? So yeah. So basically, this is literally the basic of the basic of the basic. And basically these characteristics, you literally need to know them all. And me just realize say I is missing from this list right here. I is missing, but don't worry about that. So basically, for an organism to be considered living, it needs to grow. That's one. It needs to be able to increase in size in some way, shape, or form, right? And we're not talking about like, oh, it have to grow tall like a tree, but it needs to be growing to be considered living. Then we're going to talk about reproduction. So reproduction, if something is living, it needs to be able to reproduce and produce more offsprings, right? And then I, which is missing on here, is irritability, right? So an irritability slash sensitivity. So in order for something to be living, it has to be able to respond to stimuli. For example, I'm here right now, and if somebody pinch me right now, I feel it, I'm gonna react, right? That's because I'm a living organism, right? And the stimuli is the fact that somebody pinch me, then me feel the pain. That's the sensitivity, me feel the pain, and then I'm going to respond to it, right? So that's essential for living organisms. And obviously animals might react a little bit different from human beings. Plants might react a little bit might react differently to different stimuli. For example, if you put some pesticide or maybe too much water, rain or something, as I said, different organisms react differently to different stimuli, right? And then we're going to have movement. So basically, in order for something to be constituted as living, there has to be some movement. Now, as I mentioned, movement, there's a difference between movement and locomotion. Now, we don't know if you know what locomotion is, but whether or not you know what locomotion is at this stage, you need to be able to differentiate between movement and locomotion because it's very important. When I say movement, I don't mean the thing has to get up and walk. Because we know the plants not get up and walk, right? And they are still living. So what do we mean by movement? What we mean by movement is that parts of its body must be able to move. So for example, if I am sitting down and if I can move my finger, that's considered movement, right? If I move my big toe, that is still movement. If I move my head, that's still movement. Locomotion is actually me moving from point A to point B, right? That's locomotion. So do not mix up the word movement with locomotion, even though when we think movement, we think locomotion. So don't mix them up. But basically, they just need to be able to move a part of their body, right? So if I'm sitting here, I move my big finger, that's movement. I'm doing movement right now. If I move my big toe, that's still movement. If I get up and walk from point A to point B, that's locomotion. So basically, know the difference and don't mix them up. Then obviously nutrition. Every living organism needs to feed. They need to get their nutrients from somewhere. Like how plants get their nutrients from the soil, they get their water from the soil, all that good stuff. And then excretion. Once you are living, you're eating, you're going to be producing waste. So in order for an organism to be living, it needs to be able to pass out its waste from somewhere. And for human beings, we know exactly where that waste come out from, right? It comes out through our feces, it comes out through urine, but we have to excrete, we have to get rid of that stuff out of the body. And then obviously the last one is respiration. And do not mix up respiration and breathing, do not mix them up. But basically, in order for something to be living, it has to respire. Right? And respiration, and when something respires, basically producing energy, right? So yeah. Okay, so now let's take a look at some microorganisms. The first microorganism we're going to be looking at is actually going to be our bacteria. Now, you need to know about this bacteria, and a common HSB question that I've seen is, you see this narrator I have right here, so it's a common and well-known decomposers. I have seen on the HSB exam where them ask we call it name two decomposers, and bacteria and fungus, those should be your best friends, right? Those should be your best friends. But anyways, let's talk a little bit more about bacteria before we talk about other stuff. So this is our bacteria. It's obviously a prokaryotic cell, and the reason why it's a prokaryotic cell is because it does not have a true nucleus. If you're looking at this diagram right here, we don't see a nucleus anywhere inside. There is absolutely no nucleus. What we do see is this spiral bundled up DNA, quote unquote, right? And that's the nucleoid, right? So what's going on right here is we have the nucleoid. This like a bungle up business. That's where the DNA is. So the DNA is not enclosed in a nucleus, and that is where a bacteria cell is not. It's not a eukaryote, it's a prokaryote, and prokaryote means before nucleus, right? You don't have no nucleus. DNA, the genetic information is not enclosed, it's literally free floating in the cytoplasm. So basically that's what's going on right here. Right, and the next thing too about our bacterial cell, there are no membrane bound organelles, right? And we soon take a look on our plants and our animals very soon. And if we're talking about membrane bound organelles, we know that those membrane bound organelles would be that the nucleus, it would be like our mitochondria in the case of plant cells, or chloroplast, vacuoles, stuff like that. Those are membrane bound organelles. In the bacteria, there are no membrane bound organelles in there, right? Right? So basically they are living because bacteria, they can reproduce and they can grow. So basically the process by which our bacterial cell would reproduce, so it can reproduce asexually, right? So what's gonna happen is that you see this cell right here, it would like kind of pinch off or invaginate, so basically creating two, and then it's gonna split into two. That's binary fission, right? So it's gonna create a little crease on the side, invagination, and then it's gonna split off into two. And then that's how it basically that's the main method by which bacterial cells reproduce. Now there are other asexual methods, and even I think there's a sexual method, right? But right now we cannot remember everything, but yeah. But this is the main method by which bacteria reproduces, by binary fission. So let's take a look at our bacteria cells. So what's going on here is, on here we have the pili, and basically it helps the bacterial cell to attach to surfaces, right? Because we know that bacteria, at least not all bacteria, but the general information that most people know about bacteria is that they can be harmful. Not all bacteria is harmful, but most of them are, and they can kill you. So now bacteria, it can spread, right? It can spread, and we know that antibiotics is what we would use to get rid of the bacteria. Now what's going on is the pili helps it to attach to surfaces, and obviously that's why it spread and get out of hand and all that stuff. It's very resistant. The makeup of the bacterial cell was made to resist not everything, because obviously at certain temperatures bacterial cells are going to die, but it really makes for survive. That, let me just say it like that, it really makes for survive. So our pili, we said it allows it to attach to surfaces, or ribosomes, these produces proteins. So our cytoplasm, that's the gel-like substance that surrounds not cell organelles, but whatever structures they have inside. So that's just a gel-like fluid that holds the contents of the cell, or nuclei. So basically, instead of having a nucleus with the genetic information inside, we have free floating DNA, which is the nucleoid in the case of our bacterial cell. And this diagram doesn't show it, but it usually has a plasmid, like a circular DNA free floating in it, right? So that also contains some genetic information, right? And our flagellum right here. So basically the flagellum is what helps it to move, helps it to swim, or allows locomotion, let's just say that. So it's really for movement. That's the aim of the flagella, and as you can realize, the flagella is longer than the pili, so you can see the difference in the structures. So do not think that the pili is cilia. It's not cilia. If you know what cilia is, but is pili. Pili is just pili on our bacterial cell and our flagellum, and for the plasma membrane is the same thing as cell membrane. So plasma membrane is cell membrane, and then so basically the cell membrane controls what moves in and out of the cell. So same thing. Then our cell wall, our bacterial cells have a cell wall made up of peptidoglycan. So basically that's actually a polysaccharide, right? And in the case of our plant cell, which we're not really talking about right now, but our plant cells are made up of a polysaccharide that's called cellulose, but we'll get to that soon. Anyways, it has a capsule, and basically it just helps to support the entire cell, keeping all the contents, right? And it also makes the cell more resilient. So it's like a extra protective layer, and that is why bacteria hard for dead, because they literally have three layers, right? Typical plant cell just have a little cell wall, a little cell membrane, but the bacteria say no, we need three. The bacterial cell has a cell membrane, cell wall, and a whole capsule. So that is why harmful bacteria especially can be very stubborn, because look at the structure of the bacterial cell. It is literally resilient. It's made to be resilient, right? So that's what the capsule does, and everything works together to make this bacterial cell work. And obviously, they are plentiful on the earth. There's a lot of bacteria in our mouth, upon the ground, even upon our skin. So listen, bacteria is in and on the body, right? So you can't really get rid of it. The only harm is when you have harmful bacteria taking over, because right now our bacterial cells, at least the one in our mouth and other places in our body, we have good bacteria and we have bad bacteria, and at the end of the day they are microorganisms, so it means that we cannot see them. We need a microscope to actually see these bacterial cells, because they are so small. They are so small, and they have 70s ribosomes, just in case anybody was wondering. And literally you have to look under a microscope to see them. That is why they are microorganisms, right? So for example, I'll use the example in our mouth. We have good bacteria in our mouth that obviously helps us to digest food, and basically fights off bad bacteria, right? So it's important to maintain that balance in our bodies, because that is when you got a doctor and doctor will tell you, say you have a bacterial infection. That is when the harmful bacteria get inside and start to take over. But naturally, we do have bacteria in our bodies, right? Microorganisms that help to obviously maintain the entire thing in our mouths and different places in and on our body, right? So that's just a little breakdown on our bacterial cells. They are microscopic. They cannot be seen with the naked eye. You have to look under a microscope in order to see them. And they are well-known decomposers, right? So what that means is that they can actually break down, or is that the word? Okay, they act on decaying matter. Let me just say that. So basically, as a decomposer, for example, a dog dead, the bacteria start to grow up on it and kind of eat it away, if that makes sense, right? So that's what the bacterial cell does. So it acts on dead, decaying matter. It's a decomposer. It helps it break down, right? So for example, you left the dog, you have a dead dog and dog dead today, and then by next week you look upon the dog, or not even 1 week, but you go back go look on the dog, there is no dog, the skin, everything gone, because bacteria yam up all of the skin and everything, right? So it's a decomposer. It helps to break down dead, decaying matter. Basically, that's our bacterial cell, right? So let's take a look at our virus. So now the virus has a lot of controversy associated with it. Let me just say that. So remember me in high school, and I think the teacher said that, because typically, especially when you're talking about microorganisms, you might say, oh, in biology we only talk about living stuff, right? That's what I thought too. But science says that a virus is not living. It's not a living organism, right? And the reason why it's not a living organism is because viruses do not viruses do not exist on their own. They do not have a body to occupy on their own, right? Viruses can only infect, right? They can only infect. So when they're out and in the open, so basically, there is no way to just see a virus in the air. That is what I'm trying to say. You cannot just look outside and say, oh, there's a virus in the air. No. Viruses only become viruses, right? when they are inside of a host cell. So for example, the coronavirus that did break out a couple years ago, the coronavirus would never name no virus. The coronavirus is just in the air, right? It only becomes a virus, or the only time that we can detect it is when it takes form in a host cell, right? So what viruses do, they hijack our cells. That's what they do. They hijacked our cells in our bodies, right? And they take over. Just like a parasite. Just like a parasite. When the parasite gets inside, it takes over, right? And that's why viruses are so dangerous, right? So basically, just like coronavirus, when it's just in the air, it's nothing. It only becomes what should I say? It only becomes living, quote unquote, when it gets into a living organism. So if scientists would just hold up something in the ear right now, they won't be able to tell if there's a virus in the ear or not. The only time they can pick it up is if they check a living organism. So for example, there's absolutely no way to test the ear for a virus. So when the virus gets into the body, that is when it can be tested to see if the virus is there or not. So the coronavirus, the ZikV, ChickV, all of them that did come and gone, all of those viruses, they have no power. And even the influenza, the flu, there's no way that a scientist can just take up the ear and say, oh, flu in the ear. No, because it only shows itself within living organisms. So there's absolutely no way for us to know if there's a virus in the ear just like that. The only way we can know is if we check within a living organism, right? So basically, they can only replicate in host cell. So as I said, them not nobody. They have nobody, so they can only show up themselves within a host, right? So I can be a host. So for example, me get the flu, that means that the virus gone in my body. Or if me get corona, the virus come in my body. That is why I have corona, right? The virus, right? So basically, what you need to know about viruses is that they can either have DNA, which is deoxy ribose nucleic acid, or they can have RNA, which is ribonucleic acid, right, as their genetic material, right? But never both. So there's no way you're going to find both DNA and RNA in no virus. You're not find that. It's either one or the other. No, so basically they're called a variant when they're outside of a host cell, and when they actually get into the body, that is when we classify them as the viruses, right? So the virus particles are called variant, right? They're called variant. When they get inside of the body, inside of a living organism's body, that is when we can consider them to be a a virus. So basically, that's just a photo showing the structure of viruses. And as I said before, looking at this diagram, it kind of like, I don't know if it's just me one, but it kind of resembles the bacteria cell. And the reason I mean is the layers. You see the layers after layers after layers. And not even really the animal or a plant cell and have them in layers here, like oh these viruses and these bacteria have these layers. So if I take a really good look at it, look at this, the layers. And then the virus has layers also. So why I think them have so much layers? Because they're trying to be resistant, evolution and all that stuff, survival of the fittest, because that is the whole point of being alive or being living, quote unquote, in the case of viruses. But everything they do is to just adapt so they can spread, so that they can multiply, since that is basically what they say is the goal of mankind or whatever. But every living organism or every organism, since they say viruses are not living unless they are in a host cell, is to multiply. So if we're looking here, we see this protein envelope right here. So protein envelope, and what we have going on is these are some spikes on our virus right here. So basically, we have the protein envelope, we have the spikes, we have the viral genome, and then we have the nucleocapsid, and then the viral not the viral integument, the viral tegument, basically, right? So that is basically the outline of the virus. So if you really look at it, so the envelope, right? This envelope is made up of a lipid membrane, right? That is derived from the host cell that surrounds the capsid, and basically it helps it to enter host cells, right? So basically this envelope is meant to secure the contents of the virus, if we're being specific. Because the goal at the end of the day is to multiply, is to survive, is to be resilient. So these viruses, when they're entering a host, they're ready to take over, right? So basically, their structures are meant to protect the genome, right? And that is what is going to cause the infection, right? Because we know that viruses causes infection, and we know that bacteria causes diseases, right? So basically, the entire goal of a virus is to cause infection, and as long as them protect that viral genome, which is either RNA or DNA, it will cause infection within the whole cell because it wants to take over. It's like a parasite. It is going in there to take over. So that's the whole point of the envelope, is to protect the genome inside, right? That's the entire goal of it, and the spikes. And as again, most membranes are made up of lipids, because we know that the cell membrane in our plants and our animals are also made up of phospholipids, meaning what should I say? When I say phospholipids, basically they have a hydrophilic head and a hydrophobic tail, and it's like a double membrane. So it's a fluid mosaic double membrane layer, right? So most membranes are made up of lipids that have a hydrophilic head and a hydrophobic tail. But yeah, and then our spikes, you can basically see the spikes around it, and it helps the virus to attach to host cell surfaces. So basically, I don't know if it's just me wanna pick up on one, but talking about this virus, it literally reminds me so much of the bacteria. Look at the bacteria. We talk about the pili that helps it to attach to surfaces, right? We talked about the capsule, everything for resilience. Now we'll go back to the bacteria. And it's the same thing, the envelope to protect the contents, because that is what is going to cause infection in the virus when the virus is going at the host cell, right? That is what's going to cause it. And all these spikes are going to help it to attach to surfaces. So we don't know if it's just me when I realize what I want, but these microorganisms, quote unquote, they're adapted to cause problem. They're adapted that when they go into host cells, go inside the body, human body, they must all create chaos, right? And we know that we have white blood cells and different things that can fight back. But look at this. When a virus is so well equipped, tell me now, when that virus gets into your body and basically hijacks your cells, what are gonna fight say? When these microorganisms, this virus, is so well adapted to attach to the surfaces, it's protecting its viral genome, same thing, RNA or DNA, and that is what's going to cause it. So that's the whole point of a hijack your cells and cause infection. But as you can see, this reminds me so much of the bacteria. They're so much alike. That is why they classify them as microorganisms, right? And then we said the viral genome, that's our DNA or RNA. And then the nucleocapsid is basically the nucleic acid protein assembly within the virus particle. So basically it's protecting the DNA slash RNA. So it depends on which one is in the virus, or which one is in the virus. So now the viral tegument, right? The viral tegument. We know why we have integument, I don't know, because it reminds me of something similar in biology. But the viral tegument basically also to protect the genetic information, which is the DNA or RNA, from degradation, right? And to facilitate the virus's entry into the host. So basically, all of these structures on this virus is doing everything in its power to protect the DNA or the RNA. It's doing everything to protect the viral genome, which has the DNA or the RNA. So that is why these viruses, when they get in your body, hell broke loose. Because when they get in your body, they start reproducing, they start replicating and everything. And that is why you have infection. Right? So that is why you have infection. That is why it's so important to not just look at what you can see, but if them tell us a virus not in place, believe it, because it's not gonna show up until it gets into your body. It's not going to show up, right? But every structure on this is protecting the genetic information, is protecting the viral genome, is protecting the DNA or the RNA, and that's what's going to hijack the cell. You see, as long as that DNA and that RNA is still it's still intact, it's not degraded, hell broke loose in your body as a human being if a virus get into your body. And yeah, I feel like I'm talking enough about viruses, but basically, if a virus gets, and obviously there are different types of viruses, right? But we're not getting into all of that today. So just work with this diagram of a virus. We'll just work with this one for now. Now let's move on to the fungi, right? So fungi is plural, and then fungus is singular. So we said that earlier the bacteria was a decomposer. Now your next best friend decomposer is the fungi. So basically fungi, they are well-known decomposers as well, right? So now the difference though between the fungi and the bacteria is that the fungi is a eukaryotic cell, right? And it's very much non-motile. So non-motile means that it cannot walk, it cannot technically move. So basically it cannot come out of the ground, or I don't want to use the word ground, I don't want to use the word ground, but let's just say it cannot move. Let me just say that, right? Let me just say that. So, but they are heterotroph. So yeah, they're heterotroph. So that means that they can produce their own food. No, what did I just? No, they're not. They're not able to produce their own food. Sorry. So they are heterotroph. So that means that they cannot produce their own food, right? They depend on a producer. They depend on plants, or yeah. So they cannot produce their own food. That means that they eat other organisms. Let me just say that. Yeah. So we know that they can decompose, right? So being a decomposer, it does the same thing like bacteria. It breaks down dead, decaying matter. So the same dead dog, we say the dog dead today. We go back, go look, fungus. I go start taking over and all that stuff, and we go name up, and everything. So it's a decomposer. It helps to break it down. So yeah, it's non-motile, heterotrophic, meaning that it does not make its own food, and it's also a eukaryotic cell, and which may have the actual like fungus cell, but it doesn't matter. But basically, it being a eukaryotic cell means that it has a nucleus and it does have membrane bound organelles. So if you take a look at this diagram, whenever someone talks about spores or anything relating to that, just know them about fungus, because that's how they reproduce, by releasing these spores. Then them land, and then them start grow, and then the cycle continues over and over and over again. And for the fungus, right, or the fungi, their cell walls. So they do have cell walls, just to make that here. So we know that our plants have cell walls, bacteria has cell walls, and our fungus has cell walls. No, we said that the bacteria's cell wall was made up of peptidoglycan. For fungus, the cell wall is made up of chitin, and then plants is made up of cellulose. So three different composition, although all of them are polysaccharides. Okay, so now let's talk about microorganisms and their importance. So microorganisms are very important because literally the fact that we just talk about fungus and the bacteria being decomposers, meaning that they break down dead, decaying matter, right? So when they break down that dead, decaying matter, they are releasing nutrients such as carbon, nitrogen, phosphorus back into the environment that plants and other organisms can then utilize. So for example, if you ever hear about the nitrogen cycle, then you know all about that stuff. So basically when they break down that stuff, they're releasing back nutrients into the atmosphere. Or you ever hear them put what them call the thing, not mulch, military don't remember right now, but manure. Yes, manure. That manure, as in like manure can grow plants or something like that. I don't remember how it go, but something like that would be a good example. Okay, you get nutrients from the dude or whatever because it have enough bacteria in there, and it have fungus and all kind of things. That is why it has that property. But basically, like our nitrogen fixing bacteria, yep, so it can improve soil fertility by converting atmospheric nitrogen into forms that plants can definitely use. So microorganisms are very, very important, and we're not talking about the virus that's going to cause infection. We're not including that right here. But as I said before, we have good and we have bad bacteria, right? And fungus, they also play a role in all of this. Everything plays a role, as a matter of fact. So now this is going to be the animal cell, which animal cell is a bit blurry, but we're not gonna stay long on it. We're just gonna brush over it really quickly. So don't worry about the words, but just worry about the structures. So right here we have our nucleolus. We have the nucleus. So this big yellow thing, that's nucleus. This is the nucleolus, right? And right here, this is our rough endoplasmic reticulum, our smooth endoplasmic reticulum, our Golgi body slash Golgi apparatus, our cytoplasm, lysosome. We obviously have the cell membrane, and this is the centrosome, vacuole, mitochondria, and all that stuff. So basically, we're just gonna run through the function of each part really quickly. So yeah, so we have our nucleus. We know that obviously it controls the cell. We know that part. The centrosome, we don't really have to worry about what this is. This is blue thing, but it is basically involved in cell division. So if you know the process of mitosis and or meiosis, you know what this centrosome does. But basically during cell division, there would be two of them and they separate to opposite ends of the cell. Spindle fibers would extend, the chromosomes would line up at the equator, pull them back, all that good stuff. Then we have our smooth endoplasmic reticulum, which basically is involved with the whole steroid and lipid production. Our vacuole, we know that it stores water, nutrients, that stuff also acts as an excretory cell organelle, so it gets rid of stuff out of the cell. The mitochondria, which is like the powerhouse, we know that respiration takes place within this and all that stuff. Then the Golgi apparatus slash Golgi body, that's the packaging center of the cell. Or ribosomes, they produce proteins. Cytoplasm is a gel-like substance that holds these organelles. The rough endoplasmic reticulum produces protein and all that good stuff, right? So even though this diagram is kind of blurry, you basically get the point behind it, right? So you basically get the point. So even the nucleolus makes ribosomes as well. So basically it would start right here and then they would get out to the rest of the cell, right? So ribosomes are on the rough endoplasmic reticulum and ribosomes are not on the smooth endoplasmic reticulum. Then this is the plant cell, which is a lot more clear so you can see it a little better. So the main thing about it that you need to remember is, whenever I mention, so the cell membrane controls what moves in and out of the cell. I'll just put that out there. This is the cell membrane, but here we go. So this is the cell wall for the plant, and basically plants are made up of the cell walls are made up of cellulose, right? And it's basically a polysaccharide, meaning that it's a type of carbohydrates, right? So just know that. So that's the nucleus. This is the nucleus, right? Controls the cell. Cytoplasm, that same gel-like substance that holds the contents of the cell. This is the cell membrane that controls what moves in and out of the cell. The mitochondria, respiration, powerhouse, produces energy. Then we have this extremely large vacuole, right? This is a very big vacuole. So why do you think the vacuole in the plant is so huge? Why do you think it needs this big vacuole? Like, look upon this animal cell, and the vacuole is this like a green something. Why in the plant, the 3/4 of the cell is being taken up by this vacuole? Why? Because plants absorb a lot of water, right? They take in a lot of water from their roots. They take in water, they take in minerals from their roots, and basically water is an essential part, right, of them carrying out photosynthesis, because carbon dioxide, right? So basically they need carbon dioxide plus water in the presence of sunlight and chlorophyll in order to make glucose and oxygen, right? That's the formula for photosynthesis. So they take in a lot of water, and that's why they need a vacuum. Now, why do they need a salt water? Because plant cells are taking in so much water, they don't want their cells to burst. So we do know that our animal cells, they can burst, right? Because they're on a cell wall. In the case of the plant cell, we do not want that to happen, right? Because that would be detrimental to the plant. So basically, that is why they have adapted this cell wall, which is like a structural protective barrier that keeps the cell from bursting, right? So plant cells, they do not burst, but obviously they can become flaccid, or they can become turgid. Turgid mean that they get filled with water and look healthy like this one, or they can become flaccid, or they can experience plasmolysis, where basically is where the cell membrane pull away significantly from the cell wall. That would be plasmolysis. So that's what would happen if we said that a plant cell is plasmolysed. The cell membrane will be pulled apart from the cell wall significantly, right? So now, why do we need all of these cells, right? Why we need all these cells? Because why we don't just have one cell in the body that do everything? Would that make sense? No, that would make no sense. So the reason why we need cell specialization, we need cells to be differentiated, to be different and carry out different functions. Because take for example the human body, and within the human body, a lot goes on in the human body. So look upon it like this: would it be wise if we have one cell doing everything? It wouldn't work out, because we know that we have epithelial cells, we have cardiac cells that are in our heart, we have skin cells, we have so we said epithelial cell, we have neurons, right, that go up and down, sideways, everywhere, as a part of our nervous system, right? We have a lot of cells in our bodies, right, that are specialized. We have egg cells, right? We have sperm cells. We have a lot of cells within our bodies, right? We have smooth muscle cells, we have skeletal muscle cells. There's a lot of cells in our bodies, right? So would it make sense? Would it make sense for one cell to do everything? Absolutely not. Because if you look at it like this, what do neurons do? Neurons are part of the nervous system, right? These are nerve cells, and they are what you have sensory neurons, you have motor neurons, and they work with the central nervous system, right? So you have the central nervous system, the peripheral nervous system, so basically the brain and the spinal cord, that would make up the central nervous system. And then you have the peripheral nervous system, right? With obviously would have our sensory neurons taking in the information. For example, if we touch a hot pot, taking in the sensory information, and then bringing it to the brain, to the interneuron bring it up to the brain, come back down the spinal cord, and then the motor neuron would bring it back out, to carry out the action, right? So think about it. And basically, a neuron or a nerve cell cannot do what the cardiac cell is doing. So you understand why we need cell specialization. Each cell carries out a different function, because each cell cannot do what the other one can do. And this allows for efficiency Just like if it was a business, you have everybody assigned doing a different task, it would work out much better than if you have one person running up, down, sideways, everywhere, trying to get things done. That's not efficient, right? So the goal behind cell specialization is to divide the labor and basically allows for more efficiency within cells. That's the entire role of cell specialization. That is the role. Now, we're gonna talk about these different types of cells. So basically, I mentioned them just now. Well, some of them, epithelial cell. So basically, these epithelial cell, we can find them like, what should I say, like in our skin. This is, yeah, we use that example. So basically, they form protective layers, basically covering the body surfaces and lining internal organs, right? So most organs have like an epithelium. For example, we look at the stomach, we have special epithelial cells, we have epithelial cells, and even even in the intestine. At this moment, I cannot tell you which epithelial cells are lining what. But in the intestine, you'd have cilia, right? You'd have, no, not cilia, villi. You'd have villus. So you'd have villus in the intestine, right? And you'd have a specialized epithelial cell. So now there are different types of epithelial cell. You can have the simple squamous, and they would basically look like this. Squamous, as the name suggests. You have simple cuboidal, and when we say simple, it doesn't mean that it's one layer. And then, so basically, the first part of the name refers to the layer, and the second part of the name refers to the shape. So just layer shape, and then right here, this is layer shape. So this also says simple because it's one layer, columnar because they are shaped like columns. Then you have transitional, as you can see, it starts off like this, transition to this, then you get big up here. That one is, the name basically says what it says. Then right here you have stratified squamous, where stratified meaning it's multi-layered and it's squamous cells. Stratified cuboidal, multi-layered cuboidal cells. Pseudo-stratified, where everything kind of blend up together. That's why it says pseudo. So it's not a true double layer, but it kind of appears that way, right? So that's why they say pseudo stratified, right? But basically, epithelial cells are very important in our bodies. Then we have our sperm cells, and these are basically male reproductive cells. They're produced in the testicles, and they're responsible for fertilizing the female egg. Very simple. So you have the head, you have the tail, very simple drawing or very simple diagram. Then you have this one showing the egg and the sperm fertilizing the egg in this photo. So the egg is the female reproductive cell, and it basically gets fertilized by a sperm cell in order to create offsprings, basically. No, the neuron. No, I feel like this neuron. The neuron is my favorite cell. I don't know why, but ever since I learned about the nervous system, I just love the nerve cell. I love the neuron. And basically, it is a part of the nervous system. So that's where our nerve cell is from. That's where it works. But basically, it's a part of the nervous system, and it's used to transmit electrical and chemical signals, right? Now, knowing how nerve cells work would be very beneficial and also very, very important to know how they work and how they operate and how they function, right? So basically, if we're just going to give a brief overview of it, what I would say is the dendrites are on it. So basically, the dendrites, these are basically they receive incoming signals from other neurons, right? and then it goes in. You have the cell body. That's obviously where all the cell organelles are going to be, like mitochondria and all those stuff. Then we're going to have our nucleus, and basically the nucleus is still the nucleus in every cell. It does the same thing and it controls the cell. In the case of our neuron, it is what kind of determines whether or not this neuron is going to fire. That's what the cell body, the nucleus does, sorry. So the nucleus decides whether or not this neuron is going to fire. So when neurons receive a signal, what happens is it has to meet a threshold. For example, you're in a one room and somebody had chat. Let's just say the room very noisy, right? Until you literally tone out, you're not hearing nothing. So in order for you to respond, there has to be a threshold met, right? For example, if somebody play like music really, really low, you need to hear it. It have to be loud enough for it to get your attention. So it's the same thing with this neuron. What needs to happen is when that signal comes in, it needs to meet the threshold. So basically, naturally our neurons are at rest. They're in negative 70 millivolts, and the threshold is negative 55 millivolts in order for this neuron to respond, right, or to generate an action potential. It needs threshold of negative 55 millivolts. So when the signal come in, the nucleus say, oh yeah, this reached negative 55 millivolts. We need to generate an actual potential. Right here with the axon hillock. The action potential will be generated and it run down here. And basically you have our myelin sheet or Schwann cell, which are basically like glial cells or some supporting cells, right? Yeah, so they help to produce the myelin sheet for the axon. They have the node of Ranvier. So basically, what happens with this signal is there's a thing called saltatory conduction. And that's where the signals jump. They jump in between until they get to the end. The neurotransmitters are triggered and basically they're in little small vesicles. They fuse with the membrane and get released out into the synapse. That's just a brief overview. So if you want to see the whole thing about nervous system, you can go and look at that down below. I think there's something about the nervous system there also. Yeah, I say it one, yeah. I think we did do something about the nervous system, so you can go into more detail with that one. Then okay, or muscle cells. So this is another type of cell in the human body. So these are our muscle cells and they contract in order to produce movement. So we have three types, or skeletal, or smooth, and our cardiac. So basically cardiac muscle cells, these are in our heart, obviously responsible for pumping our blood around the body. So very, very important. In our smooth muscle cells, these are responsible for involuntary movements within the body, right? Involuntary movement. Involuntary meaning something like breathing. You don't have to think about breathing. You don't have to think about even seeing. You just see. You just get up with a morning time you just see. But the skeletal muscle cells, these are voluntary movement, meaning say if you get up and say, oh, I'm gonna walk, these are the cells that move your foot when you walk. So this is voluntary. This is 100% conscious when you're talking about these muscle cells, right? These skeletal muscle cells. As voluntary, involuntary, heart. Just remember that. Then we have different transport across the cell. So diffusion. So essentially, diffusion is the movement of particles from an area of high concentration to an area of low concentration until it is very much evenly distributed, right? So this is our phospholipid bilayer. This is our cell membrane that we're looking at right here with our hydrophilic heads, hydrophobic tails, and basically most substances can pass through via diffusion, right? I was said before that the cell membrane controls what moves in and out of the cell. We know that. Now, those that can pass through are able to diffuse across the membrane, meaning that they leave the high concentration and they go into the cell where there's a low concentration. So that's basically how diffusion works. Or for example, you're in a classroom and somebody come in here with KFC, and then in a one side of the room, you up on the other side, the scent gonna literally travel to you and you're gonna smell the KFC. That's how diffusion works. Then you have osmosis, and osmosis is very similar to diffusion, right? So it's very similar to diffusion, except it's happening in water, meaning that um specifically moving water molecules from an area of high concentration to an area of low concentration across a semi-permeable membrane until it is evenly distributed. This is the biggest difference between I would say diffusion and osmosis, is that one, it's happening in water, and then two, it is going across a semi-permeable membrane, as you can see right here, but it's the same concept, high concentration to low concentration, right? And then I just realized that looking at this diffusion, I forgot one other important thing to mention. So this right here that we're looking at is simple diffusion, right? Now you need to know that we have simple diffusion and we have facilitated diffusion. So not all molecules are going to be able to move across the membrane. So you see this cell membrane right here we're looking at right now, not all molecules are going to be able to diffuse freely. Like these ones that's coming out of the cell, not all of them. So what's gonna happen is that in facilitated diffusion, certain particles, let's just use glucose for example, cannot just diffuse across the membrane just like that. And the reason why they cannot do that is because um, of the hydrophobic tails. So not everything, and the fact that these tails are faced inwards, it means that certain things cannot interact with them, right? So you don't put, it's like putting dry and water. So if the head them tell us that them hydrophobic and them like water, they're going to be outside. If inside the tails are hydrophobic, meaning that they don't like those stuff, it cannot pass through. What it's going to have to do is to use like a carrier, for example, like a you call it like a carrier protein or a channel protein, something of that nature, in order to get it across the cell membrane. And if you're wondering what that looks like, it would probably look like a hot dog, something like that. Yeah, something like that, like a hot dog to get it across the membrane. No, for active transport, this is the most different out of all of the transports across the cell membrane, and it's mainly because it uses ATP. So, simple diffusion, facilitated diffusion, and osmosis, all three did not use ATP. The cell can just do it. You don't need no energy. For active transport, there has to be an input of ATP, aka energy, right? Adenosine triphosphate. Yeah, there has to be ATP. So in active transport, particles are moved from an area of low concentration, so low concentration, to high concentration against against a concentration gradient. Sorry, so low concentration to high concentration against a concentration gradient, right? So that's the biggest difference, because in diffusion and osmosis we said that high to low. In active transport, we're saying it's low to high. So you see that it's reversed, and we're also using ATP. And in this, it's like a pump, right? So it's like a little protein pump. So what's gonna happen is it's going to close here and then open, grabbing the molecule. And so close here, open, and release it over here. So it's like a little pump in order to pump the molecules inside the cell, and it's the same thing. So active transport is kind of similar to facilitated in the sense that we need the protein to move it across the membrane, right? So like a protein pump, right? And then we said that before in our facilitated diffusion, can use a carrier protein, can use a channel protein, you have options, right? You have options. Now let's talk about our process of photosynthesis. So looking at photosynthesis right now, we need to know the formula. That's very important in order to understand what's going on. So what's going on up here? This is carbon dioxide plus water, and this should be sunlight over chlorophyll, so light energy meaning sunlight, and then chlorophyll, which is inside of our green plants in the leaves, and all of that is going to combine to produce glucose and oxygen. So this formula is the formula for this glucose. So this is C6H12O6. This is glucose. 60, not 0, 6O2. This is oxygen. Now this 6 and this 6 and this 6, they're all just balancing the equation. So technically you can dash them off if you want, right? But this is how a balanced equation for photosynthesis would technically look, right? So this is the process now, the process by which plants use sunlight, carbon dioxide, water to produce glucose and oxygen, and it occurs in the chloroplast plant cell where light is absorbed by the chlorophyll. So chlorophyll is like a green pigment within our green plants, basically. Now we have two stages. We have our light independent reaction, and then we have our light dependent reaction. So basically, this is the light reaction, meaning that it is light dependent. Then the dark reaction does not require light, and basically it means that this is independent. So it's light independent over here and light dependent over here. For the light reaction, aka light dependent, meaning that you need light to go on, it's going to involve both photosystem 1 and photosystem 2. We're going to involve photolysis, which means splitting of water, and then oxygen is going to be released. And then the light dependent reaction takes place within the grana of the chloroplast, and basically the grana is made up of stacks of thylakoids. So they are like these discs, they're circular disc-like things, disc-shaped things, right? That's what make up the grana. It's a stack of thylakoids. And then ATP and NADPH are going to be produced. For the light independent reaction, no photosystem is required. Photolysis, meaning splitting of water, does not take place, and carbon dioxide is absorbed, right? And then you're going to have the light independent again occurring within the stroma. So the stroma is basically the space within our chloroplast. It's not the grana, it's not the thylakoid, it's just the little empty space on the side, right? So that, and then we're going to produce glucose in our light independent reaction. Okay, so now let's talk about some factors that affect the rate of our photosynthesis. So basically, if we go back to the formula, we said it's carbon dioxide plus water, sunlight, or chlorophyll, right? So it means that any of those things can affect the rate of photosynthesis, right? Because all of them are needed in order to drive the process of photosynthesis. So the light intensity is very important. So the higher the light intensity, the more the rate of photosynthesis will speed up to a certain point, and we'll talk about that soon with a diagram. So it will increase until it cannot increase no more, until it levels off. So as I said before, too much light can damage plant cells. You have CO2, which CO2 is needed to drive the process, so it can also become a limiting factor. Temperature also too. So we know that with plants, there's a lot of enzymes involved in the process, right? There's a lot of enzymes, a lot of proteins, a lot of good stuff. And proteins get denatured at high temperatures, right? So we need the optimum temperature for the plant in order to drive photosynthesis. If it gets too hot, a problem. If it gets too cold, it's also a problem for the plant. So yeah. So and also the water availability and then chlorophyll. So the more chlorophyll, the more light it can absorb and all that stuff. So if we take a look at this diagram, we're seeing that rate of photosynthesis, we're seeing light intensity, right? So here we go. Light, the more light you visualize, increase the light, it's going to increase the rate of photosynthesis until a certain point where it levels off. That would be the point where photosynthesis cannot go any faster, it cannot increase anymore. And this also means that something else has also become the limiting factor, right? So it's either wanting to increase, even though it's getting enough light, it gets short on carbon dioxide or gets short on water, something else start to limit it and it cannot increase past this rate. And the same thing for carbon dioxide. You keep on increase the carbon dioxide and it's good, right? Until it starts to levels off. And that means that something has become the limiting factor here. Now with temperature, it will continue to increase, get to the optimum, and then proteins, enzymes denature at extremely high temperatures. So it's gonna fall right here, and denature means that they're going to break down or it just cannot survive basically, right? It's going to drop again. Now let's talk about nutrition. So basically it's where plants obtain nutrients and then convert it into energy. That's basically what nutrition is. And it's essential for the human body. We need it to function properly, gives us energy, functions in growth and repair, rebuilds tissues, a lot of stuff. So our carbohydrates, they provide energy, proteins, build and repair tissues, our fats also provide energy. Vitamins, minerals with what we would consider micronutrients, meaning that we need them in much smaller amounts, meaning that we just need a little bit to survive. While carbohydrates definitely a macronutrient, we need a lot. How we use energy a lot. So we chat a lot, we move a lot, we need energy. And also a lot of proteins for our teeth or hair, a lot of stuff we need it in order to build and repair tissues, right? And then obviously water, we need water a lot in order for hydration and also for our body to function optimally. We need a lot of things. So we have different types of nutrition now. We have heterotrophic nutrition, and basically these organisms do not produce their own food. They depend on, I would say, basically they feed on other organisms, which can either be the autotroph, meaning the plant, or it feed on other animals, right? So in the case of humans, we feed on everything. We eat the producer, the plants. We eat everything. We eat animals, we eat plants, we eat everything. Then you have autotrophs, and these are basically these are plants, basically anything that can photosynthesize, and some algae can actually photosynthesize. So basically, what happened in here, these autotrophs, they can produce their own food. They can utilize the energy from sunlight because they have the structures for it, and they can make their own food, and that's how they live, and that's how they survive. And in fact, they are what continues the entire the food chain begins with producers. And it goes to the consumers, primary, secondary, tertiary consumers. Then you have saprotrophic nutrition, and that would be like a fungi or bacteria, as we said before, that they can actually feed on dead decaying matter, so they can break down things. So that is what saprotrophic nutrition is. They feed on dead decaying matter. And parasitic nutrition, and it literally means that they obtain their food from a host, usually causing harm. An example, parasites like tapeworms, but even thinking about the same virus, it causes harm as well, right? And then we have symbiotic nutrition, where they live together and exchange nutrients, right? So the type of symbiotic relationship that would result in mutual benefit would actually be a mutualistic symbiotic relationship, meaning that both organisms benefit, they live together and they exchange nutrients, as in you do something for me do something for you. Then you also have commensalism, which basically commensalism is where one organism benefits without harming the other one, right? So it means it means that me go benefit and nothing I got to you just today, right? Me not hurt you, me not do anything, but me wanna go benefit technically, right? Without hurting you. So it's one organism benefit without hurting the other one. So right. So then we have parasitic symbiotic relationship, which basically is what we kind of already described here with the organism obtaining their food from a host and then causing it harm, right? So those are different types of symbiotic relationships. Now let's talk about diseases that you can get when you have improper nutrition. So malnutrition, and it's basically also known as undernutrition. So it's basically caused by a lack of nutrients, either poor diet, not eating enough, not having access to food, and it's basically if your body cannot absorb the nutrients from food properly, that can also cause malnutrition. So then obesity, so it's basically having too much body fat, anorexia nervosa, and then you have um bulimia nervosa. So basically these are two eating disorders that are very popular. In the anorexia nervosa is basically where it's an eating disorder, but it's also a mental health condition where it's basically the person starve themselves in order to keep their body weight low. So basically people like this would usually struggle with obviously body image issues, meaning that they would starve themselves, they just want to be skinny, so they starve themselves. So that's basically anorexia nervosa. While bulimia nervosa is basically where the person binge eat like stuff on their TV, and it's like kind of like trying to deal with their stress or their whatever or insecurities, and they just eat, and they will just eat a lot, like as in out of control a lot, just them everything. And then after them done eat everything, them start to feel very disgusted with themselves and they start to vomit it back up, right? Because they're very much weight conscious, self conscious, everything. But it's like as I say, it's a mental kind of like a mental disorder because it's all in the mind. It's all about body image that definitely affected as well. Why they act like that, and after them don't eat everything, it's like you buy 10 patty, eat it, so don't eat it, and you deal with your problems. And when you're done with it, you realize, oh, well, and they me just put on weight, and then you already struggle with your body image, right? So it's like binge eating, yeah, right? So binge eating can lead to bulimia nervosa, right? And when you're done, you try to vomit it back up. So basically, these are two very popular eating disorders that are very prominent today. Then you have kwashiorkor, which is a protein deficiency. So if you ever see like them pictures with some little pitney, and this is not only for kids, but if you ever see some little pitney where them belly that just hang out, like as in you can see them rib cage, and then you just see them belly swallow. So that would be the kwashiorkor that them have. And then you have marasmus, which is another protein deficiency where them just dry out, them just look maga, dry out, skinny. So basically, kwashiorkor and marasmus, these are both protein deficiencies, but their symptoms are different, or the way they look on the outside is different. Kwashiorkor, you can pick it up from a mile away because them belly, even though they're so malnourished, you can from not having enough protein, them belly just big. And then marasmus on the other hand, they're not a big belly, but them just very dry out and very underweight. And even the person who has kwashiorkor, they're also underweight too, but them just have the swollen belly as a characteristic. Let's just say that. So now we're going to talk about food chains.